Hello, I'm Matt Kennedy.
And I'm Chandra Tucker. We're associate professors at the University of Colorado School of Medicine, Department of Pharmacology, and the senior authors on a collaborative project developing novel optogenetic approach for inhibiting synapse function.
Tools for switching off neural activity have been extremely powerful for defining the specific populations of neurons on the brain that drive behavior. The past decade has seen an explosion of new genetically encoded molecular tools for precise regulation of neural function. We developed a new tool for local, inducible, long-term suppression of neural function that works by disrupting release of neurotransmitters required for communication between neurons. Neurotransmitter release is mediated by the SNARE protein family. Clostridium botulinum neurotoxins are protease enzymes that specifically cleave SNARE proteins and potently block neurotransmitter release. Direct application or genetic expression of these toxins has been a classical approach for constitutively silencing synapses, but currently these reagents do not afford spatial and temporal control of synapse function.
We decided to update these classical reagents using optogenetic approaches. We focused on botulinum toxin serotype B, which cleaves VAMP2 on neuronal vesicles. To regulate the toxin with light, we first split the protein into two halves. We attached the two protease halves to light-induced dimerizers. These proteins have low affinity in dark but interact with high affinity in light. We tested Cry2, SIB1, and eyelid SSPB blue light-induced dimerizers. We wanted to generate a protease that was inactive in dark but could be functionally reconstituted in the presence of light. We tested different sites to split the protein in half, settling in residue 146. When the protein is split at this site, the fragments could self-assemble even in dark. To disrupt the dark assembly, we mutated residues at the interface between the two fragments. A mutation at Y365A resulted in a protease with low cleavage activity in dark and high activity upon light exposure, as shown in this Western blot. We moved this version forward into neurons.
Hi, I'm Brooke Sinnen, one of the co-first authors. I wanted to test whether photoactivatable botulinum toxin could cleave endogenous VAMP2 in neurons. To do this, we transfected cultured hippocampal neurons with the toxin, along with M-cherry to fill the cell, and synaptophysin GFP to label presynaptic terminals. We measured VAMP2 cleavage with an antibody that recognizes the full-length, but not toxin-cleaved VAMP2. While we observed light-activated cleavage of VAMP2, it was relatively slow, taking several hours. To improve kinetics of the tool, we directed the toxin to synaptic vesicles by fusing one of the fragments to synaptophysin, which resulted in much faster VAMP2 cleavage following light exposure. This amount of cleavage also disrupted synapse function, measured by recording miniature postsynaptic currents.
Given the promising results in neuron culture, we next wanted to test if an in vivo introduction of the toxin could disrupt synaptic transmission in an intact circuit. I injected adeno-associated viruses encoding each half of the toxin into the CA1 of mouse hippocampus, then 2 weeks later prepared acute slices and recorded evoked synaptic responses in subiculum. We observed a robust, light-dependent reduction in synaptic currents within about 15 minutes following light exposure.
In order to show that the photoactivatable botulinum neurotoxin can be used in an intact living organism, we expressed it in motor neurons of the nematode C. elegans, because they are important for normal locomotion behavior. So if you put the worms into liquid media, they show this typical swimming behavior that can be recorded using this optogenetic imaging setup here that allows simultaneous blue light illumination. Indeed, what we saw is a 50% reduction in swimming frequency after 1 hour of blue light stimulation caused by photoactivatable botulinum neurotoxin activation.